We'll be talking about DNA as the genetic material in this video. But before moving on to the experiments that proved that DNA is the genetic material, let's first go back to historical perspectives. The first question is, what is molecular biology? So, molecular biology is a science that helps us understand life at the most fundamental level. That is, the study of biology on a molecular level, including structure, function, and makeup of biologically important molecules like DNA, that is deoxyribonucleic acid, RNA, that is ribonucleic acid, and proteins. Now, the science of molecular biology began in the 1930s after the work that was done by Gregor Johann Mendel, Thomas Hunt Morgan, and the chromosome theory of inheritance that was put forward in the 1910 by Sutton and Boveri. The term molecular biology was given in 1938 by Warren Weaver. Warren Weaver was actually a mathematician, and he named this science as molecular biology. And this field involves many other areas of biology, such as biochemistry, genetics, physical sciences, and chemical sciences. In the 1940s, relationship between genes and proteins was put forward by Beadle and Tatum. Now, around 1869, Johann Friedrich Miescher, who was a Swiss medical student, isolated an acidic substance from pus cells after treating these cells with pepsin. His discovery was published in 1871. He called this acidic substance as nuclein. He found that this substance was rich in nitrogen and phosphorus. The term nuclein was changed and nucleic acid term was given in 1889 by Richard Altman. In 1919, Phoebus Levene stated that nucleic acid are made up of three components. They are pentose sugar, nitrogenous bases, and phosphoric acid. Units are joined together to form a polynucleotide strand, and these three components join as units, which are called nucleotides. The first X-ray diffraction pattern of DNA, which showed a regular structure of DNA, was given in 1937 by William Astbury. In 1952, Erwin Chargaff stated certain rules which told us about the complementarity between the nucleotides, that is, the pairing between purines and pyrimidines, the molar composition of bases in the DNA strand. The first experimental evidence which proved that DNA is actually the genetic material was given in 1944 by experiments that were conducted by Avery, MacLeod, and McCarthy. So in 1944, the first experimental evidence came that cleared that it is DNA which is acting as the genetic material and not proteins. A confirmatory experiment which confirmed that DNA is the genetic material was performed by Hershey and Chase in 1952. In 1953, the structure of DNA for the first time was published by Watson, Crick, Maurice Wilkins, and Rosalind Franklin. 1961, Jacob Monod gave the operon concept and also gave the name messenger RNA for the intermediary molecule which is acting as a messenger between DNA and proteins. Between 1961 and 1965, genetic code was cracked, the relationship between DNA and proteins, how the DNA expresses itself was given. Now, how do we define gene? Gene is a physical and functional unit of heredity that carries information from one generation to the next. The DNA sequence necessary for the synthesis of a functional protein or RNA molecule is a gene. Gene was first detected and analyzed by Gregor Johann Mendel and subsequently by many other scientists. Mendel stated that physical traits are inherited as particles and did not know that these particles were actually chromosomes and DNA. Subsequent studies showed the correlation between transmission of genes from one generation to the next and the behavior of chromosomes during sexual reproduction, that is, during the meiosis and fertilization. These and related experiments provided a strong evidence that genes are located on the chromosomes. Now we say that DNA in most of the cases is the genetic material. Where DNA is not present, their RNA acts as the genetic material. So there has to be some criteria which DNA is fulfilling so that it is acting as a genetic material or it is qualifying to be the genetic material. The first criteria is that it should be capable of storing all the information about structure, function, development, and reproduction in all living organisms. And it should be capable of transmitting this information faithfully from one generation to the next. This function of the genetic material is known as a genotype function. The second function of the genetic material is that it should be capable of expressing. The genetic material must control the development of the phenotype of the organism, a virus, bacterium, plant, or animal. That is, the genetic material must dictate the growth and differentiation of the organism from single-celled zygote to the mature adult. This property of the genetic code is its phenotype function. So a genetic material should be capable of storing all information about structure, function, development, reproduction in all living beings, and it should possess the power of accurate replication so that the daughter cells possess the same genetic information as present in the parent cell. The third criteria is that a genetic material should be stable and at the same time it should be capable of change, it should be capable of modifications by mutation and recombination so that it can contribute to adaptation and evolution. The chromosomes are composed of two types of macromolecules. One, proteins, and second, nucleic acids. Nucleic acids are of two types, DNA or deoxyribonucleic acid, RNA or ribonucleic acid. For many years, there were considerable disagreement among the scientists as to which of these macromolecules, whether it is protein or it is nucleic acid, that is carrying the genetic information or that is acting as the genetic material. So during the 1940s and early 1950s, experiments were carried out which clearly showed that it is the nucleic acid which acts as the genetic material and not the proteins. So these experiments specifically showed that wherever DNA is present, DNA will act as a genetic material. If DNA is not present, like in case of certain viruses which have RNA, their RNA will act as a genetic material. Now, initially, scientists thought that DNA was a very simple molecule and it will not be able to carry out all the genetic information. Therefore, till the beginning of the 20th century, proteins were regarded to be the main hereditary material. So the reasons why proteins were regarded as a main hereditary material were, number one, proteins at that time were known as very large complex macromolecules. And if the macromolecule has to store all the information, then it has to be a very large molecule and it has to be a very complex molecule. The first point that was given was that proteins are known to be very large, complex macromolecules, and therefore they could be the ones which are carrying all the genetic information. Second point they gave was that proteins are the most abundant in the body. So scientists at that time said that proteins are very large, complex macromolecules and are most abundantly present in the body. Therefore, they could be the ones which are acting as the genetic material. Third point that was given was that proteins are functionally the most diverse macromolecules which are known. That is, proteins form the structural framework of the cell. They are associated with the performance of all sorts of body activities. They have a regulatory effect. Therefore, they are functionally the most diverse molecules that are known. Fourth point that was given was that proteins are structurally the most diverse macromolecules. So proteins, they said, occur in varied forms. The possible combinations of 20 amino acid in proteins can be 20 to the power n. So if you have a strand of polypeptide which has 10 amino acid, the number of combinations can be 20 to the power 10. So 10 amino acids can combine in 20 to the power 10 combinations, and therefore they are most structurally diverse molecules that are known. Then certain indirect evidences were given that proteins could not be the genetic material. It could be DNA, which is the genetic material. So certain indirect evidences were given. The first indirect evidence that was stated was that DNA is located in the chromosomes. So most of the DNA of the cells is located in the chromosomes, whereas RNA and proteins are also abundant in the cytoplasm. Second point that was given was that a precise correlation exists between the amount of DNA per cell and the number of sets of chromosomes per cell. Third point that was given as an indirect evidence was that the molecular composition of DNA is mostly the same in all different cells of an organism, whereas the composition of both RNA and proteins is highly variable from one cell type to another. Fourth point that was put forward as an indirect evidence was that DNA is more stable than RNA or proteins, which are synthesized and degraded quite rapidly in the living organisms. Since the genetic material must store and transmit information from parents to offsprings, we might expect it to be stable like DNA. So these correlations suggested that DNA is the genetic material, but they didn't prove it. Proof came by certain experimental evidences, and those experiments were performed. And the first proof that came that DNA is a genetic material was in 1944. So in the next lecture, we'll be doing the first experimental evidence that proved that DNA is the genetic material. Thank you.